you have learned the biosynthetic pathway of glycoproteins. As far as the degradation pathway is concerned, it's not very different as compared to the degradation you have learned in the case of proteoglycans, or specifically GAGs. One of the most important factors here is the hydrolysis which takes place in lysosomes, because you know that these proteins are directed towards lysosomes, where they are then acted upon by many enzymes for their degradation. These enzymes are specifically, or most of the time, are the exoenzymes, that is, they digest or cleave the most terminal part of the residue or the protein, glycoprotein first, and then they gradually go on cleaving the terminal parts. So in this way, we can say that there is a gradual reduction in the size of the protein because of this gradual cleaving. And then they also, the second part, one is the enzyme action takes place on the terminal part, and second is that it removes the different residues in a reverse order as compared to when you compare it with the synthesis of the protein. So the residue which is attached or added to the glycoprotein at the very last stage, it is the first which is to be removed. In this way, the degradation of the glycoprotein is very similar to the degradation you have learned in the case of proteoglycans. If any enzyme is missing which is involved in this degradation, then definitely it will lead to not only the problem in this degradation, but it will also have medical implications, which you will learn later. A little bit of the overview of certain diseases I will talk now, but then later we will talk about these diseases in some detail. There are some rare autosomal recessive diseases which are caused by the defects in the enzymes which are involved in the degradation of glycoproteins. What is meant by autosomal recessive? It shouldn't be difficult for you to know, if you have a little bit of background of genetics, that if a gene which is encoding those enzymes, it is located into autosomal chromosomes, and it is the inheritance is of what we say the recessive fashion. It's you know what is meant by recessive gene, a gene which is only showing its expression or its phenotype when it's in homozygous condition. So they are rare autosomal recessive diseases. They leads to the partial degradation or impaired degradation of these glycoprotein molecules, and as a result of this, what will happen, that lysosome will be accumulated with these degraded undegraded molecules. One example is alpha mannosidosis type 1. As the name shows, that alpha mannose is not degraded because of the defect in the mannosidase enzyme. It leads to immune deficiencies. It is diagnosed through certain enzyme assays, which we will see later. The main thing you should keep in mind, that one of the common thing the GAGs and the glycoproteins which they share with each other is the way they are degraded, and the major enzymes which are involved in these degradations are the lysosomal enzymes.